Hi, year 12s, recapping our previous video. We looked at the mitochondria here and the different anatomy of it. Obviously, the important parts are going to be the phospholipid bilayers, the orange and the yellow there. All right, the intermembranous space in between, the cristae folds, and of course the crystal matrix. In our previous video, we looked at glycolysis, which occurred outside of the mitochondria. All right, again, we also have the Krebs cycle and the electron transport chain, which are the three interconnected biochemical pathways here. All right. And again, yeah, we looked at glycolysis, so that's the splitting of glucose, of course, occurring in the cytosol of the cell. So splitting of glucose into two 3-carbon molecules from a 6-carbon glucose that are called pyruvate. And we'll touch upon that in a moment. Again, our outputs and our inputs here. All right, inputs glucose, 2 ATP, 1 coenzyme NAD plus. So 2 of those molecules. Outputs 2 pyruvate, so splitting that glucose, 4 ATP, making 2 net gained ATP because minus 2 plus 4, and then 2 NADH coenzyme. All right, we're looking at the Krebs cycle now. All right, essentially we're making more coenzymes. All right, more coenzymes so that the electron transport chain can work effectively. All right, so let's talk about pyruvate. So obviously that's what's made via the glucose there. Okay, so these are the 3-carbon molecules. So pyruvate then has an intermediary step, is what I like to call it, but it's also called the link reaction, another name. And that's where this pyruvate is converted into acetyl-CoA, all right, which also makes more NADH. Carbon dioxide also a output of that, but again, we don't really need to know this process, but just be aware that pyruvate gets turned into acetyl-CoA, all right, in this link reaction. Cool, so we have the Krebs cycle. All right, so what does it do? It generates a lot of high energy electron and proton carriers, but mainly, really, the electrons are the important part. Well, the secondary purpose is these hydrogen atoms, which once they become H+, become those protons, okay, as they split from the coenzymes. So what do these two things do? They're used up in the electron transport chain. Obviously, they carry electrons, right? So they're used in the electron transport chain. So what else is happening here in the Krebs cycle? Carbon dioxide molecules are a byproduct, so they're going to be released. All right, and again, a small amount of ATP is now created, so 2 ATP molecules produced. Something to note is that this happens twice. All right, so we have 2 pyruvate molecules, remembering that we got 2 from the splitting of glucose, which then gets converted into 2 acetyl-CoA. So this happens once, and then it happens again. All right, so that happens 2 times. All right, hence it's the cycle. And where does this occur? The location here is that mitochondrial matrix, so the inside of the inner membrane of the mitochondria. So we're looking at our inputs and outputs now of the Krebs cycle. All right, again, the location of the mitochondrial matrix here. So inputs, 2 molecules of acetyl-CoA, 2 ADP and inorganic phosphates. All right, that's what that PI stands for there. Inorganic phosphate. 6 NAD and then 2 FAD. Okay. What are your outputs? 4 carbon dioxide molecules, so why we exhale. 2 ATP is king, so that's energy. And then, of course, our electron carrying, proton creating coenzymes, NADH and FADH2. Again, the numbers are not really important except for what's starred there, which is ATP. All right. Please watch this video if you haven't had a chance. It is hilarious. Thanks for watching, guys, and remember to study this hard. And we're moving on to the electron transport chain in the next video.